First of all, I want to thank the organizers for getting me here, and I want to apologize actually for chilling your nerves in coming really in the last minute, but Lufthansa, Charles de Gaulle and Paris traffic did their part of this, so sorry for that. So, and I want to thank actually also Manfred for giving a nice introduction into the skin. Is this working? Yeah? No? Ah. Okay. Hello? Hello? No. Now it's working. Okay. Okay. So we had already some talks about skin here, and skin is not only the organ of our body where vitamin D is synthesized. It has many other functions which are impaired during aging. And in fact, many studies also on vitamin E supplementation in elderly have the problem that in elderly persons, the vitamin D activation due to UV light is also impaired due to changes during the aging of the skin. So this skin aging, or big parts of our body, is actually catalyzed by the impact of the environment, and the biggest impact is generally acknowledged to be the sun. And if you look for the sun spectra here, you have roughly the UV part of the spectra, the visible part, and the infrared part of the sun spectra. In terms of total energy input on our body, if we are sun exposed, about 50% of the energy is coming from infrared. About 8 to 10% is coming from the UV, and the remaining of is from the visible spectra. This is a result actually of the Earth's atmosphere, and if you looked in our cartoon here a little bit, that big parts of the UVB and essentially all UVC is filtered by the atmosphere, is never reaching us if we are located on the surface of the Earth. Visible UVA, in fact, the visible spectra is penetrating the atmosphere, whereas infrared is somehow filtered by the water, which is in the upper parts of the atmosphere, and which is called water filtered infrared, which we are exposed normally here on the Earth's surface. So our cells in our body are all exposed to all these different irradiation parts of the sun spectrum, and in general it was accepted that in our cells are chromophores absorbing this energy from the irradiation. Many of these chromophores are activating, forming various oxidants and reactive oxygen species, and then you have a number of adverse effects which in general contribute to the skin aging and to premature skin aging. However, it seems not to be so easy because, it's hard to see here, but here you can see different parts of the sunlight penetrate to different depths of the skin. So in general, UVB is absorbed in the very, very upper layers in the epidermal layer of the skin. UVA is penetrating into the deeper layers, into the dermis, and infrared is penetrating through the dermis and perhaps even deeper. So this gives the different effects of this irradiation. While UVB is largely connected, or UVB damage is largely connected to the formation of skin cancer, of various types of tumor changes in the skin, this is connected to the upper layers here in the epidermal thing. UVA was, and there are many, many studies out, is connected to something which is basically not so dangerous, but this is wrinkle formation. Wrinkle formation is actually catalyzed here in the dermal layer of the skin by the fibroblasts sitting here. And about the effect of infrared, there was for a long time no discussion in the literature whether infrared has any effect on skin cells at all. So we tested actually in a test tube the effect of this different irradiation types on a protein solution. And we looked for the inactivation of GAPDH. And as you can see, UVA, UVB, and for comparison here, gamma irradiation readily inactivates the enzyme, whereas infrared, regardless of the dose, does not do anything. So it seems to be a type of irradiation we are very resistant. This inactivation of the enzyme is in general connected, you can look here, with the oxidation of the enzyme. Here we measured protein carbonyl formation in gamma and UVB. Very dominant, you find a little bit in UVA, some bands are here, nothing in infrared. However, if you expose cells to these irradiation, the picture might look different. And we started our experiments with a series of questions where we actually address the question whether there are additional chromophores in cells, more than in proteins, in isolated proteins, and what is actually the contribution of UV and infrared to skin damage or to damage of dermal skin cells. First of all, let me show you some of our data about the role of UV, and in particular, since we are in the deeper layer, or we use fibroblasts, the cells of the deeper layer of the skin, we are referring here to UVA. So the question is, and it's known for a long time, that there are chromophores for UVA. The major oxidant species formed there is singlet oxygen, which is activated here, and then a number of different oxidation-related processes are shown. Whenever you take a proband and irradiate parts of his body with UVA and measure afterwards protein oxidation, you see something like this upper picture here. So without irradiation, basically there's no oxidation. If you damage them with UVA in this case, you find all what is green is protein oxidation. And if you look 24 hours later at the same spot, you find a picture like here. As you can see, the very upper layer, the stratum corneum, which are essentially the dead cells, they are still oxidized. Then you find a layer here in the skin, which is not oxidized anymore, 24 hours after exposure, and the deeper layers are still oxidized. So this is essentially, which is the skin layer coping very well with the oxidation process and with the protein damage process. This is the epidermal layer. And as we are working on the degradation of oxidized proteins, and we heard a little bit from Irfan today already about this, this is also the layer where in the cells are massive concentrations of the proteasome, which are related to the degradation of oxidized proteins. The deeper layers, however, have less proteasome in the dermal layer, there are less cells, and a lot of these oxidized proteins are persistent for a long time. And if you think, I am as a German, think about a vacation, going to Italy or to Spain, going into the sun today and tomorrow again. So if I go tomorrow again, I have still this damage from the day before. So it will accumulate in repeated doses over the time and essentially lead to the skin damage. So actually the same you can see, it's difficult to work with human probands here for a long time. So we repeated the experiments in vitro in a cell culture. UVA damage leads to protein oxidation. Fibroblasts have, of course, proteasome, but what is interesting to us is that UVA irradiation led to a decline in the proteasomal activity. Somehow, dose dependent, you see more or less it levels off. You find after a certain level a decline or no proteasome activity anymore, whereas the proteasome is still there. So it's just inactivated. And we tested what is the reason for that, and a couple of years ago already we published a number of data showing that the proteasome is inhibited by protein aggregates. We could find in these cells an increase in protein aggregates, so actually UVA is leading to an aggregation of proteins, and we think that these protein aggregates are declining or reducing the activity of the proteasome. One of the major highlights of skin aging or premature skin aging is the overexpression of the matrix metalloproteinase 1 and the decline of the tissue inhibitor of the matrix metalloproteinases. We can find this also in our model. And to our surprise, somehow it's coincidences that at the same doses where the proteasome is low, where you have lots of oxidation of the proteins, the MMP1 expression is very high. Therefore, we address the question whether this is somehow connected, whether both events might be connected somehow. And we used a different model. We gave protein aggregates to the fibroblasts. Fibroblasts are able to take up these protein aggregates, and as you can see here, over time, it takes a couple of days here, 2 weeks in this sample. These protein aggregates accumulate in these cells. The proteasome activity is going down as expected, and to our surprise, also the MMP1 expression goes up and the TIM1 down. So you don't need the UVA to induce these changes, just the prevalence of protein aggregates is leading to that. And of course, there is a more easy way, an easier way to inhibit the proteasome, and that you just use proteasome inhibitors. And we used lactocystin, and also just if you block the proteasome, the MMP1 expression goes up. So the proteasome, as an enzyme responsible for the degradation of proteins, is able to induce something in the cell. And of course, this is a question, and we asked for the mechanism, and we came up with a hypothesis or theory here, and tested for the major contributor to MMP1 expression, which is the c-Jun, or AP1 transcription factor, and the component c-Jun. And you can see, regardless in which way you block the proteasome, you always see a higher c-Jun in a cell. And interestingly, also in the case of lactocystin, where there is no oxidative stress, at least, you find also increased level in phosphate c-Jun. And you could show that the transcription factor is going into the nucleus, and this all expression of MMP1 was blocked by Jun kinase inhibitors here in this one. We came up, after all, with a hypothesis how we think this is working, or UVA in this case is working. I told you that the proteasome is responsible for the degradation of oxidized proteins, but many tend to forget that this is only one function of the proteasome. The proteasome has another major function, the turnover of all proteins. And one of these proteins is the Jun protein. It's shown that it's degraded by the proteasome. So the Jun, if you reduce Jun, less Jun can become phosphorylated. So in a kind of way, the proteasome is blocking the activation of Jun by the Jun kinase here in this case, and therefore it's counteracting the activation of AP1. And in a situation where if you have oxidized proteins, the proteasome is blocked, this blockage here, this inhibitory pathway is blocked, you have more Jun, more phosphorylated Jun, and then you have a higher transcription factor in this case. That was actually the data, or the example of data I wanted to present for the UV. And now I want to come up with a total different mechanism, which is related to infrared. Infrared is a difficult thing, because there's no known chromophore in the cell. We don't know. Infrared is generally absorbed by water. If it's absorbed by water, it forms heat. So in the literature, if you look for the devices used normally in the literature, you have a problem. Because if we modulate the impact of heat, the impact of energy in such a petri dish, which you can see here, drawn on this cell, and you give the usual used irradiation doses, you find a high temperature. Or what is red here is above 42 degrees. So essentially, if you use models for infrared, you boil your cells. And yeah, if you boil your cells, they form oxidants with time. So it was a very long, and this, you can see it very nicely. It's a mathematical model modulating the temperature, and in such a petri dish here. And actually, we did the same. We used thermosensory elements and measured the temperature. It's true. So the model is really working. Essentially, what you have to come up with is an experimental condition where you find that the temperature in the water cooling device is so that it really has only minimal changes in the layer where your cells are sitting. And actually, what is really crucial, and we developed such a device here, is that the petri dish which you have here has direct contact to the water cooling here, the water cooling system. And in most cases, if you use cooling plates or temperature plates from metal, this cooling is not efficient or sufficient to maintain the temperature here. And then we thought, is there really an oxidative damage due to a water-filtered infrared A spectrum here? And we challenged actually the model which is in the literature, where it showed that infrared is somehow absorbed by mitochondria and forming ROS. So this is a model which was present in the literature until 2 years ago, and we thought maybe it's just the heat, and the heat is activating somehow the metabolism, acting on the mitochondria, and then you might have ROS and radical formation at this point. And we did a very simplified approach. We used this infrared without sufficient cooling, temperature homeostasis with infrared, and just a heat exposure or heat shock, and looked for the effects. What you can easily see, you form a lot of oxidants. This is a DCF fluorescence, just independent, dose dependent on infrared if the cooling is not sufficient. But the same amount you form also just by temperature. No irradiation. It's just 42 degrees here in this case. If you make the cooling sufficient here in this case, no increase. We were not able to find an infrared A-induced oxidant production if the temperature is really maintained at 37 degrees. We did the same with another parameter. We measured oxidized proteins. Heat, again, is increasing oxidized protein dramatically until some experimental points we couldn't do because the cell was just dead. So we didn't find cells anymore. The same is with infrared without sufficient cooling, again, oxidation, and no oxidation if you do the cooling properly. So it seems to be that, and we published this last year, that the major effect that infrared has, and what was studied in the literature, is due to heating effects for us. So, but what is the role of mitochondria in the system? And we tested this, and we blocked mitochondrial respiration here in this case for a long time, so that mitochondria are not able to produce many oxidants at least. And we found anyway also with cooling some oxidation processes here. But if you compare it here at the same doses, you have a twofold increase. In the not blocked mitochondria panel here, you see a sixfold increase. Indeed, mitochondria are a major contributor to infrared-induced radical formation or oxidant formation, but this is the mediator of this, we think, is now the heat. And if you look for the infrared, the infrared spectrum here, for the absorption, you see that many cellular components, like the cytosol, like the cell membranes and so on, absorb major parts of the infrared here, and this is due to the water content in these cellular fractions, because water is one of the major components of the cell, and this will absorb a lot of this infrared-related energy and lead to cellular effects. So whatever we can say now, and I think this is a working current model we have, infrared is interacting with the cellular water, producing heat, and via mitochondria then producing reactive oxygen species. So what I wanted to show you as a conclusion now for my talk here is that irradiation, solar irradiation, can lead to skin damage via totally different components. This is, in the case of UVA, it's a direct protein oxidation and aggregation, or in the case of infrared, via interaction with mitochondria, heat formation, and or with water, heat formation and mitochondria, leading actually to the same readout that we have, but the mechanism is totally different. Thank you for your attention.
Thank you, Tilman, for this nice overview of the effect of not only UV but again infrared spectrum. So now we have time for questions. I may start about UVA.
Yeah.
So when you are talking about UVA direct effect, do you mean direct effect of photosensitization reaction in order to promote the degradation of protein?
I think you need some photosensitizer, but you have the photosensitizer in the cell. You have a lot of cofactors. The B vitamins, for example, are essentially very good chromophores or sensitizers for the UVA.
So
yeah?
you try to see action spectrum in order to better identify putative photosensitizer?
We haven't done it, but it is then done. It's basically the flavonoids in the cell, the FMN and FAD, which are in this part of the spectrum major absorbers and sensitizers.
So you think that again is a main component?
Yeah, it is that we could show clearly.
Thank you for this nice talk. Concerning the RAS production and the heat, do you have any evidences that your mitochondria are somehow uncoupled, so they need to work faster to maintain the membrane potential, and then this could generate gas?
Not yet. Okay, we are working on this. It's interesting. There seems to be some components of the respiratory change absorbing infrared, but it's a minor part of the energy input of the total absorbed infrared. So we are trying to triggering this out, how mitochondria react during heat, during infrared, or whether there are differences in the uncoupling, in the throughput to the respiratory chain, ATC. So we try experiments on it.
Okay, please.
Hello? Yeah.
Yeah, so you said that when you have the protein aggregate, you get upregulation of MMPs. Beautifully you showed that. So that means that we can safely say that the aggregation of proteins, upregulation of MMPs can be directly, potentially related to aging of skin.
We think yes. Yeah, it's let's say there are numerous studies out on UV-induced skin damage. Almost every of these studies is referring to DNA damage. Well, lots of things if you find in the literature, but we think this is just due to the fact that protein damage is not as often investigated. So I think it's a major contributor because due to the absorption spectrum which you have, this is not, we are talking about UVA, right? That's above 320 nanometers. So there's DNA not absorbing. Nucleotides have no impact on this. So that we think protein-related damage and in particular protein aggregates are important.
And what about the exposure to heat in sauna? I was planning to build a small sauna at my home.
Okay.
So should
I think this heating experiment are difficult. Sorry. What we are doing here, this is tissue culture. in a human being, you have numerous physiological regulatory mechanisms limiting the heat, right? And so you'd never reach in a human being in deeper skin layers 43, 44, 45 degrees because you have the blood circulation which is limiting the temperature. In our studies, we have also done studies with humans in sauna. You have an increased surface temperature increase to 41 maybe degrees, but the total body temperature in sauna is not really increasing. You are talking here the whole body average blood whatever temperature. You are talking 0.3 degrees. So it's not really that we are talking here about the same levels as in an experiment. The problem, what I really wanted to show that this is some of these experiments or data are purely due to an artifact of the experimental setup, because you induce a heat which you never reach in vivo, right?
Very nice talk and very clear. You focused your talk on collectivism if you want, but to my knowledge, one be wrong. Gelatinase and plasminogen activators are very, very important for degradation of matrix by fibroblasts. So do you have any data on that?
Not yet, but as you can assume, we are now looking for different genes upregulated by the inhibition of the proteasome or by the induction of protein aggregates. So I don't know whether these gelatinase, which is MMP3, I guess, right? MMP2, okay. So whether this is coming up, I don't know. So, but these experiments are in progress still. Okay?
I think we have a final
Have you identified the aggregates that are responsible for inhibition of the proteasome activity? In an earlier study, we reported in a model system that it was lipofuscin. Have you any information on that?
We didn't find lipofuscin because it's a single dose irradiation. If we do repeated doses in a cell, we find lipofuscin. So I think a single treatment is not necessarily leading to the final formation of this big aggregate, which is lipofuscin. So I think this is our initial protein aggregates which we can find with radio labeling methods. So, but in repeated doses we find clearly lipofuscin that's formed.
Okay, I think we will close the session. So first of all, thank you for your